I wanted to welcome everyone to this evening's program, and thank you all so much for turning out for Nature of Seeing, Art, Perception, and the Brain. My name is Mike Morawski, Director of Education and Public Programs here at the Portland Art Museum. And before we introduce tonight's distinguished speaker, and tonight's program, although advertised originally as a little bit of a panel discussion, we have been working so closely with OHSU and David Wilson, our speaker for tonight, and he has plenty to be able to share through all the work that he's doing. So we went ahead and just have David presenting tonight instead of having a panel discussion, which we thought would be a program that would be perfectly suited to the audience and the projects that we've got going on. So I have no doubt you will be thoroughly thrilled by the program this evening, but I did want to make that note in case you're thinking, where's everybody else? Because we've had a lot of partners in this program, and I'll definitely thank them before we get started. But before I introduce David, I want to get a little bit of context for sort of why we have this program and we've been thinking about neuroscience and the brain here at the Portland Art Museum. This program tonight fits into a larger initiative that we're calling the Nature of Seeing, and that's part of the exhibition that many of you possibly have seen upstairs called Seeing Nature, Landscape Masterworks from the Paul Allen Family Collection. And first of all, I think I speak for the entire museum staff when I say how honored and thrilled we are to be able to host this special exhibition of these works, the first time many of them have been seen in public before. And in thinking of how to frame this exhibition, aside from the story of a deeply passionate collector or the deep art historical stories around each of these paintings, we were sparked by something Paul Allen wrote for the catalog when we were planning this exhibition. And he said, "People talk about how our brains are wired to see landscapes, to look at landscapes and to see what's going on in them. So there's something about landscapes that seems almost universally attractive." Combine this spark of Paul Allen's quote with the fact that he's been a driving force in supporting neuroscience research, including founding the Allen Institute for Brain Science in Seattle, and the fact that Pacific Northwest has truly become a neuroscience powerhouse, to use the words of Congressman Earl Blumenauer. So we decided to go ahead and take this angle with the Seeing Nature exhibition. And we've been so fortunate to have partners like OHSU to work with us and to support this project and this exhibition throughout. In the exhibition galleries, you'll see a small space dedicated to exploring this theme, the nature of seeing, including a video produced for this project by the Allen Institute for Brain Science. Additional content and resources were supported and provided for us by OHSU, as well as partners at Northwest Noggin, which is a neuroscience outreach and education group based here in Portland. They're doing fantastic work in schools all across the area. So special thanks to OHSU's Brain Institute, the KCI Institute, the Allen Institute, and Northwest Noggin for supporting this project with us. So I'm pleased to introduce David Wilson, who will be speaking tonight. David is the chair of the Department of Ophthalmology, director of the KCI Institute, and director of the Christensen Eye Pathology Laboratory at OHSU's KCI Institute, as well as the Teeley Petty Chair in Ophthalmology at OHSU's School of Medicine. You can see why we have him speaking tonight. He's received his undergraduate degree in chemistry from Stanford University and attended medical school at Baylor University, where he graduated with honors. I told you I'd go over your whole bio, David. David has been on the faculty at OHSU since 1988 and became chair of the Department of Ophthalmology and the director of the KCI Institute in 2005. His areas of medical interest are cancers of the eye, gene and cell-based therapies for inherited retinal disorders, and eye pathology. On a warmer and much more personal note, David has a lovely wife, Nancy Green, and 3 children, Reed, Eric, and Claire, and he wanted to share with me that he enjoys the outdoors, cycling, golf, tennis, and running, and so I hope you enjoy getting to know him better this evening. Please join me in giving David Wilson a warm welcome.
Well, thanks. It's really wonderful to be able to speak here, and thanks for such a nice introduction. Now, for really hundreds of years, artists and scientists both have been very interested in the visual system and how it works, really the topic of this talk, the nature of seeing. And I have to say I'm astounded at the number of people here for the talk, with the ducks playing and the Timbers having a playoff game and Thursday night football, it really, really very clearly shows that there is great interest from everyone really in how art and science of vision are developing. But I did think because of Paul Allen's great interest in art and his great interest in science, but also his great interest in sports, that a good way to do this presentation would be as a world championship of visual art and science. So I've assembled two teams, and they said it wasn't going to be a panel discussion, but I've really got some pretty good panelists here. So on team art, I've got Monet, Klimt, Signac, Picasso, Chagall, Renoir, and O'Keeffe. Now these are really all household names, or at least in this house. This is the Portland Art Museum, and several of these people have exhibits upstairs. And now on team science, maybe not so well known. We've got Ramon y Cajal, Hubel and Wiesel, Yarbus, Livingstone, and Sachs. Now, most of you probably haven't heard of these people, but I will tell you within that group, there's 2 Nobel laureates, or 3 Nobel laureates actually. So they're not quite as much of an underdog as you might think. But I do think that the other side has the home field advantage here. So now this is an audience participation lecture too, and so you've been given 2 cards as you came in. One is blue and one is yellow. So maybe you can hold those up so we can see if that's going to work for me here. Oh yeah, that's great. It's kind of like a punctillious painting. It's really great. Yeah. Okay, well, so the way we're going to do this is we're going to have a series of questions, and since it's not really an athletic competition, when we go through the questions, whoever you think has done better at explaining the question, that's who we're going to have you vote for. So the first question I put to the groups was, what does art teach us about the visual system? And so Team Art is going to take this one first since they're the home team, and they chose Picasso to respond, and he decided that he was going to show a video to aid his response. So hopefully this will work. So this is a time-lapse video of Picasso painting. Maybe some of you have seen it before, but it's quite fascinating, and this is his description, because he's a panelist, right? So it says, I start off by using lines of high contrast. I find this is easiest for the visual system to organize the work. Then I began to add detail and color. And now I'm going to advance this a little bit because, great art does take a little while. So now he has added detail and color. And as I add detail and color, these elements will replace most of the black lines, which really do not exist so much in nature. The contrast between the color works just as well. Finally, I'll mix up some of the elements. So let's see, advance to that. I'm going to mix up some of the elements because I find that the visual system finds that more interesting than a truthful rendition of nature. Very interesting. So Picasso has brought up that art teaches us about contrast, detail, and color. And then he's thrown in this little thing at the end about mixing things up to make it more interesting. So pretty good answer. The artists are pretty clever guys and gals. Now we'll have the scientists respond. Now this scientist I'm pretty sure nobody will ever heard of, A.L. Yarbus. A.L. Yarbus is a Russian psychologist in the 1950s. He was very interested in how our visual system interacts with the visual environment. And he studied this by doing tracings of eye movements and looking where the eye movements went. And he actually was very interested in art, and so he would do these tracings over art. So A.L. Yarbus is going to give the response for the scientist to that initial prompt. And here's one of the paintings from upstairs, Klimt's Birch Trees, beautiful painting. And what Yarbus says is that I have studied this by doing ray tracings, and you can see the reflections on the subject's eyes there, and that's the way you do these tracings, or one way to do it. For instance, here with the subject looking at the beautiful Klimt work, Birch Trees, you can see that the eye is drawn to areas of high contrast and detail. You could see those tracings on the tree there. Also, color is of great interest. You look at the blue flowers. And it seems that this birch trunk in the left-hand portion of the painting is of particular interest. So he's also thrown in something that maybe we can talk about later in the lecture that might be of particular interest. Okay, so that's round one. So we have both the artist and the scientist responding. So if you think the artist responded better, hold up with the blue card. And if you think the scientist did better, oh, the scientists are really getting snuffed here. Okay. Well, not too surprising, not too surprising. Okay, well now before we go on to the next prompt, I need to teach you a little bit about light and the visual system. So, what we think of as light and is actually a very tiny part of the electromagnetic spectrum. So I think even way back in evolution, the rest of this had been reserved for the internet, Facebook, and cable TV. Yeah. So the question is, why did organisms become sensitive to light? Why were they interested in light? And there's some really great properties of light that make it attractive for organisms to engage. And that is that it's very fast and it can travel for long distance. So it can be a great way to get information from the environment. But the question then becomes for these single-celled organisms and the primordial ooze, how do I do that? And that's where vitamin A comes in, because vitamin A can exist in two conformations. And when it absorbs a photon of light, it switches from one conformation to the other. And that little switch in conformation can be triggered to produce vision. So now if we fast forward billions and billions of years, we have this very elegant structure, the human eye. And the way the eye works, of course, is that, using the pointer here, if it'll work, light comes in through the pupil, goes through the cornea. The cornea is the clear, fairly sturdy tissue in the front of the eye, and it does most of the focusing of light. Then the light goes across a little chamber of fluid, and it encounters the lens. And that fine-tunes the focus, and then the light goes through this vitreous gel to engage or be absorbed in the retina. Now, if we look at the retina, and if we look at it about 1,000 times magnification, we can see that it consists of a lot of layers of cells. And this layer here is the one that absorbs the light. And if we look at that at maybe 10,000 times magnification, we can see this layer of this photoreceptor. And then maybe at 100,000, what we see is that the photoreceptor has these layers of membranes. And they're very regular. And that's where the light is absorbed. Because if we could look at it at 1,000,000 times, what we would see that is within those membranes, there's a molecule or many molecules of vitamin A. And when that vitamin A molecule absorbs that photon, that little switch that we saw back in the single-celled organism triggers an electric impulse or a neural impulse that goes from the photoreceptors through the retina and eventually goes back to the brain through the optic nerve. So that's the chemistry that triggers our vision. So now we can go to the second round as to how does the eye work. So now most of you will never have heard of Ramon and Cajal, but he's one of my heroes. He's really just an amazing person. He wanted to be an artist when he was growing up, but his dad wouldn't let him. His dad wanted him to be a barber, which looking at him is kind of ironic. But he persisted, and he actually became what is perhaps one of the greatest neuroscientists of all time, and in fact is known as the father of neuroscience. But he really took advantage of his great artistic skill, because what he did was he made very intricate handmade drawings of neural networks. And those neural networks were what let him determine how the eye worked in many ways. He found these cone-shaped cells, which he knew were the ones that were sensitive to color and to brightness of light. And he found these other more rod-shaped cells, which were not as sensitive to light and were better set up for motion. So what Cajal knew was that the center part of our retina was the part that we would use for detail and color. and that the peripheral part of our retina would be used for motion and less sensitive to light. So this question was kind of right in the wheelhouse of the scientist, right? I mean, this is pretty basic stuff. So how are the artists possibly going to respond to this? Well, you might be surprised. So the artist's response is going to be given by Renoir. The question to him is, how did the eye work? And he says, well, artists have developed a very sophisticated understanding of how the eye works. He says we didn't always understand that. And if you look at this painting, which is upstairs, thanks to Paul Allen and the Portland Art Museum, this one by Bruegel the Younger, all of the detail, the whole canvas is covered with the same amount of detail. And when you look at it, your eye just kind of goes very comfortably around the painting because everything looks about the same detail. But in Moran's painting of Venice, there are some differences. Our eye is really drawn to this bridge in the foreground because it's much brighter and there's more detail. Similarly, the Ducal Palace in the background is brighter, and even though it's distant, there's quite a bit of detail. And of course, in Renoir's painting, there's great detail in the face and the eyes, whereas the periphery is much less detailed and even not completely focused. So he knew that if he would paint with detail, that the center portion of the eye would focus on that part of the painting. So the artists are quite aware of that. And this was hundreds of years perhaps before the scientists got to it. So very interesting that they've been able to do the same thing. So, okay, so that was round 2. Let's see who's going for the artist for this one. Wow, boy. Okay, it's closer, but fortunately I'm not blue-yellow colorblind. I'd say that's almost a toss-up, Okay. Okay, well now it's going to get really a bit complicated here because the next question is a really, really hard one, and people are still working on it. And so the question is, how is the image in our eye perceived by our brain? So since it's so hard, they're going to work as a team on this one. You can see Team Arts group there, all familiar names. And the Team Science may not be quite as familiar, but Hubble and Wiesel won a Nobel Prize. You may have heard of them. They're really the pioneers of the neuroscience of the visual system. Margaret Livingstone is a neurobiologist at Harvard, and you may have seen her book in the gift shop. She's really written some really great stuff on vision and art, and much of what I'm talking about tonight is from work that she's done. Now, you may not have heard of Oliver Sacks, but he also has a book there, and he's was a wonderful person. He died just a few months ago, ironically, from cancer of the eye. But he's going to participate on this team nonetheless. And so we're going to go through this maybe a little faster, but if there's questions, we can cover them in the end. So the scientists are going to go first, and they go back to Descartes. And Descartes says, well, I think the eye works like a camera. And so he decided that he would dissect an ox's eye. And so what he did a careful dissection so that the back of the eye was almost transparent. And then he put it on a shutter in a window, and he observed the back of it. And what he could see was a formed image of the external environment. And so he was right. The eye does work like a camera. But then this question, though, is how does it get from the eye to the brain? Well, Descartes thought that there was a little man living inside our brains, and that he was watching our eyes, and that was how things worked. Well, not quite right. But Hubble and Wiesel really are the ones that on this panel came up with the way that it works. So light comes in through the pupil, goes through the lens, and gets absorbed, as I showed before, in these photoreceptor cells. Now, there's a lot of photoreceptor cells, but then the impulse from these goes through some intermediary cells and then goes to what's called a ganglion cell. And the ganglion cell sends a fiber back to the brain. Now, every ganglion cell represented here has several photoreceptors that lead to it through these intermediary cells. And some of those photoreceptor cells are going to stimulate the ganglion cell, and some of them are going to inhibit it. And whether or not an impulse goes from the ganglion cell to the brain depends on the balance of the stimulus and the inhibition. So now this explains a very interesting phenomena that you may have seen. This is a grid, and most people looking at this grid will see some dancing black dots. And what that is where those dots are, the cells that are responsible for that receptive field are receiving more inhibition from the grid, from the black parts, than they are stimulus. And the dots dance around because as we move around, that part that's being inhibited changes position. So that is a very interesting representation of these receptive fields. Now, what Hubble and Wiesel discovered really is that if you start putting these cells together, you can start creating complex patterns. Like this might be a cell that receives this input only responds to an oblique white line. And you might be able to build another field where instead of just responding to white, it responds only to red color. And so you can build more and more complex patterns based on this type of network. And with all the billions and billions of cells in our eyes and brain, you can even build cells that only respond to a face or only respond to movement. And so what's really happening is our eyes are breaking down the elements of the external world and sending that back, and the brain is reconstructing our visual space from the information that it's receiving from the eyes. Well, I wish that was the end of the story, but it's even more complicated. So this is work that Margaret Livingstone has really written some very nice stuff about, that there's really two visual systems superimposed on one another in our brain. There's what she's called the where system, which is a more primitive system that's responsible for motion, depth perception, and spatial organization, and segregation of the figure and background. Now, interestingly, this system is color blind. It doesn't see color, but it's very fast. Now, the what system recognizes objects, face recognition, and color perception, but it's very slow. And these two systems are in different parts of the brain, which is helpful for the scientist, as you'll see in a minute. So this is a picture of me doing my best Greg LeMond imitation. I used to say Lance Armstrong, but I don't anymore. And you can, if you take this image in Photoshop, you can kind of simulate what happens with the what and the where system. So the where system is very sensitive to motion, and so it can detect and accurately record the motion. But remember, it's black and white. That's all that it's going to see. Whereas the what system sees colors, but it's slow, and so it's going to see blur where there's motion like this. Now, the what system can be broken down further into two other systems. Surfaces with color are extremely low resolution for the what system, and so you see just blurred color things. But things with a lot of detail are very accurately depicted by the what system. So these are our two visual systems. Well, the skeptics out there might say, I don't believe it. How can that possibly be? How would you know that? Well, that's where Oliver Sacks comes in. Because he was a neurologist that had the most amazing practice where he would see patients with very selective strokes that affected their visual system sometimes. So what he was able to describe were injuries, bilateral injuries to these different systems. So here's a patient with bilateral injuries to the where system. So they really have trouble orienting things in space. So this patient has drawn two renditions of a bicycle. So you can see all the elements, but they're all in the wrong place. This is a clock, and this is a cross. And then there's the motion part of the where system, and this is a description of a patient that has deficits in the motion part. She would see a prolonged motionless image and be visually unaware of any movement around her, though her flow of thought and perception was otherwise normal. For example, she might begin a conversation with a friend standing in front of her, but not be able to see her friend's lips moving or facial expressions changing. And if the friend moved around behind her, she might continue to see him in front of her, even though his voice now came from behind. Or she would see a frozen arc of tea coming from the spout of a teapot, but then realize that she had overfilled the cup and that there was now a puddle of tea on the table. So these things really go right to what was being described as these two different systems. Similarly, with the what system, this person was asked to name these objects on the left, but could not name them. But she could perfectly draw them, but couldn't name them, unable to recognize those. Here's a verbal description. I can see the eyes, nose, and mouth quite clearly, but they just don't add up. I like this one. At the club, I saw someone strange staring at me and asked the steward who it was. You'll laugh at me. I'd been looking at myself in a mirror. All right. So one of Oliver Sacks' books is titled The Man Who Mistook His Wife for a Hat, which is a great title. So his books are very interesting, and you might want to check those out. So there's also the color system, and you could have a selective defect in the color system. Now, this is 3 renditions of a painting. This is the painting, the normal painting, and this is the rendition of the painting by someone who's colorblind, just typical color blindness. But someone with bilateral injuries to the color portion of their visual system cannot see any color and can do the form portion of the drawing, but can't do the color. And here's the description from Oliver Sacks. He was profoundly disturbed by the wrongness of the appearance of everything he saw. Even his dreams were gray and drab. Foods looked disgusting to him, and so finally he began to eat only achromatic foods like black olives, white rice, black coffee, because they appeared relatively normal. So quite a disturbing thing. So that's what the scientists think about the way that we get visual perception from our eyes. Now, again, you might think the artists are really going to have a hard time responding to this, but you might be surprised. So the artists say, well, what took you so long? We've known that forever, right? We've done these drawings where we just kind of put in the form like you see in the what system, and the color doesn't really matter because you just told us in the color in the what system is kind of messed up, and we've been doing that. See, we knew that. Or here's a painting by Chagall, and he really almost makes no attempt to get the color within the structures, but that really doesn't bother us. We're able to understand what the artist is intending there. So artists have really taken advantage of a very sophisticated element of our visual system. And that's really, that's part of why these paintings, these pointillist paintings work, because the color part is fairly low resolution. And so the colors will blend before we lose the forms that contain the colors. And that goes right with the way the visual system works. Another interesting thing is if you look directly at the reflections of the sails in the water, you'll say, well, yeah, there's lots of dabs of paint. But if you direct your gaze toward the rocks, your peripheral vision, which is lower resolution, will fuse those much more readily. So artists have really understood many of these same concepts in their paintings that only now we're beginning to understand from the neuroscience. And now we're back to Picasso, who said it's more interesting to mix up the painting, right? Well, isn't it remarkable how the painting that he did resembles this abnormality of the Ware system in the patient? So now I'm not saying that he had that, and of course not, but artists recognize that things that really tweak our visual system are interesting, and that's something that the brain is going to enjoy, and that's the way that they explore the way the visual system works in art. Now this is pretty complicated, but I think it's really interesting because it goes to the same concept of the Ware system. Now if you remember, the Ware system is colorblind, and so it only detects luminance, how bright something is. Now, in this ray tracing, remember there was a lot of interest in this trunk. And if you look at that over here, it's very easy to see the trunk. It does really kind of engage the eye. But if you look in the grayscale of that so that you've removed the color, it's curious that Klimt has made this base of the tree almost exactly the same as the background. So that really confuses our WEAR system. We don't really know where that trunk is with our WEAR system, and so it really engages our eye, and it makes that trunk much more of visual interest than other aspects of the painting. So that was the third prompt. So now it's a real test for the audience to see, were we going to give that one round to the artist, or are we going to give it to the scientist? Okay. Wow, it's still pretty close, but I think maybe the scientist just edged him out just a little bit. Maybe we gave him too much time at the mic or something, right? But still, I think we probably have to give the edge to the artists, especially since they're the home team, and we always want the home team to win, right? So, okay, now, like any good event, there should be an epilogue or interview with the stars of the game, and so I thought we might have one of those here because it's another very engaging thing as to artists that have abnormalities of their visual system. So another painting that's upstairs is one of Monet's water lilies, and this was painted according to the label up there and in the program in 1919. And at that time, it's very well documented that Monet was almost legally blind from cataracts. And so he's pretty good, even with cataracts, not bad. And that's a picture of him in bed after cataract surgery, which back then patients would be in bed for 2 to 3 weeks with cataract surgery. Now it's like a 30-minute operation, so a lot of advances. But he was very reluctant to have his eye surgery. And you can read what he said about his vision and prior to his cataract surgery. He says, "I no longer perceived colors with the same intensity. What I painted was more and more dark, more and more like an old picture." And of course, cataracts serve as a filter and really change our perception of color. He actually, for a while, struggled after his cataract surgery because he had trouble matching colors on his palette that he had previously mixed. So down below is Gustav Klimt, who based on studies of photographs is felt to have had strabismus or crossed eyes, which is really kind of interesting because if that's true, he didn't have normal stereo vision. But if you look at his painting, there's very clear perception of depth in that, and there are many ways that you can perceive depth even if you're monocular. So you wouldn't expect someone just because they had crossed eyes to not be able to paint with depth. In fact, Margaret Livingstone feels that many artists don't have normal stereo vision and that it's actually an advantage for them. So another very common condition is macular degeneration, and Georgia O'Keeffe had very severe macular degeneration by the time she died. Of course, she lived to be in her 90s. The painting that's upstairs was painted well before she had any impairment of her vision, but later in her career, she used assistants to help her with her painting, and she would direct some of the paintings, but they had much less detail than her early paintings did. And of course, Degas is also felt to have had macular degeneration. He was a painter early in his career, but because of his loss of vision, was not able to do that later on. But he could still use his visual memory and sculpt because that he could use his tactile sense and still create things that he was able to create in his visual memory. So I think that may be the last prepared slide that I have. Yes, it is. So, but I want you to do something for me because I want to thank the Oregon Brain Institute, who's really responsible for these great programs that address how science interacts with so many things in our lives. But in particular, I want Bobby Haggerty to come up here. Bobby, please. Where are you? And so what I want you to do, Bobby is really responsible for how wonderful the Oregon Brain Institute is, especially the interactions with the community. And So what I want you to do is hold up both your yellow and your blue, and we're going to take a picture. You're going to be facing the other this way. Yeah. So back up a little bit. Not so tall. So if you get a nice picture, can you have this off, the screen off? Great. Yep. So look at the camera. Yeah, this is great. So Bobby's going to be retiring, and so we're, we have a wonderful person replacing her, but Bobby, it's really been wonderful. Thanks so much for doing that. That's really, I hope the picture will turn out. So I also want to thank Paul Allen and the Portland Art Museum. This is a wonderful, wonderful exhibit. It shows so many great paintings, but also now maybe it helps us engage as to how the visual system works. And I really want to thank Christopher Howell, George Petrusek, and Stephanie Ryan, who are people that work at the Eye Institute, who helped me put this presentation together, which I never would have been able to do with my limited tech skills. So, and thank you all very much for coming. Maybe you'll get to see the exhibit and still have time to watch the ducks or something else, right? So thanks so much. So I'm happy to take a few questions if people have those, and then maybe we won't take too many so that people that want to can get up to the exhibit. There's somebody in the middle there with the yellow, and they have to give you a microphone because they're recording this, and without the mic it won't record.
In black and white. Why was there an association between damage to the visual system and damage to what we can imagine?
Yeah, I think that's a really excellent question. And I don't know that I can really say that. I certainly don't know why there might be the memory of color might be absent. I actually think based on what he said, that he definitely had some memory of color because he was so disturbed by the absence of it. But why that wouldn't show up in his dreams may speak more to the way that our images are creating in dreams, which I really don't know the answer to, but that's a really wonderful question. Yes, front row.
What prevents motion sickness from looking at paintings?
What? Oh, so if you get motion sick from looking at paintings, that's interesting. Well, that's actually pretty similar to say I'm a pathologist as well, and so look at a lot of things through a microscope. And usually there's someone that's with me, that's learning, with me on how to do that. And it's very, very common for them to get motion sickness because as I move the slide around, it's very disorienting. And so I think the same things that would work for other types of motion sickness would probably work for that type of motion sickness. But of course, no, I don't think so. I think it's of course, it's very simple. You don't have to continue your gaze, so you can just stop, right? Yeah. Yes?
You mentioned Monet's paintings after cataract surgery. I've seen a great number of his very late paintings, which are wild with color.
Right.
Quite unlike the serenity of his other landscapes. Could it be he was overcompensating in some way, pretending stuff? I don't know. But it's quite unlike the kind of taste he used previously.
Yep. Well, I don't know if that was evolution of what he wanted to do with color or whether that reflected the change in color that occurs with cataract surgery, which it definitely does change your color perception because you've essentially removed a yellow filter from with the lens. Yeah.
I think we had a question, a couple questions in the back.
Okay, we'll take a couple more, and then maybe I'll happy to stay around up front and answer questions too.
Well, thank you. I was wondering if you know of any landscape artists that have documented color blindness of one sort or another, just straight color blindness, where they don't see one color and how they deal with that.
Yes, I'm afraid I don't know of any landscape artists that are colorblind, but of course, people that are colorblind do see some color. It's just a different representation of color than is present in people that are not colorblind. Yep. One more question, maybe? Yep. Yes.
I've often wondered, with reference to perception of motion, whether the human eye and/or brain perceives motion as a series of discrete frozen frames, such as in a movie reel. Is the production of motion pictures as we know them an imitation of the biological process of seeing motion in the natural world?
Yeah, it's a great question, and the answer is no. That's not the way we detect motion. We don't see it as a series of snapshots that we put together. And it goes to what I very briefly mentioned there, is that our brains are actually reconstructing the visual space. And you can imagine that it would be extremely disorienting if we were taking snapshots and had to reassemble what was happening, because our eyes move incredibly quickly. And we would not really be able to assemble all of those images. And so what's really happening is our brains are creating the visual space, and it's doing it based on the input from the eyes, but it's not taking frame shots that are put together. Okay, well, why don't we stop there, and people can enjoy the exhibit, and I'm happy to stay here and answer some more questions. Thank you.